Good morning, and we'd like to welcome you to our webinar today on particulates and their role in the medical device industry. This webinar is aimed toward companies that develop such products or manufacture cardiovascular medical devices or devices that are likely to come in contact with a patient's bloodstream. The FDA has frequently requested these devices to be tested for particulate matter. Here to present is Ryan Lunsford, one of our resident experts here at Nelson Labs. Just for information in the future, if you miss one of our webinars or want to refer back to it, you can find them on our webinars page under the Knowledge Center of our website a few days after the webinar finishes. You can receive notifications and other testing news or updates by becoming a fan of Nelson Labs on our Facebook page or following us on Twitter. Just as a way of some Nelson news, we have dramatically decreased the price of our seminars for all the remaining events in 2011. Check out the new pricing on our seminars page. We have also improved our sample submission form, so please go on to nelsonlabs.com and get the new form. As for the webinar, we welcome your questions, and you can submit them at any time. We will randomly answer as many as possible during our last 15 minutes. Now, let me tell you a little bit about Ryan and Tanya, our presenters today. Ryan Lunsford, he received his Bachelor's of Science degree in biology from Utah Valley University. He has been with Nelson Laboratories for over 5 years as the Particulate Study Director. He has written many unique procedures during that time and has participated in thousands of studies. He frequently consults with sponsors, often with the FDA involved, to discuss and aid in test development. Ryan is also CQIA certified. He was published in the May 2009 issue of MD and DI magazine with an article on particulate testing titled Analyzing Particulate Matter on Medical Devices. Tanya Morris has been with Nelson Laboratories for over 18 years, and she is our pharmaceutical section leader that oversees the particulate testing. She is a specialist microbiologist with the National Registry of Certified Microbiologists. She is an active member of the AAMI committee that wrote AAMI TIR 42, Evaluation of Particulates Associated with Vascular Medical Devices. We'd like to welcome Ryan and Tanya, and the time is now theirs.
Thank you, Mike. So today's presentation is geared toward particulate matter testing specific to medical devices. The point of this presentation also is to share some knowledge that we've gained in the last several years, well, several decades actually, of particulate testing here at Nelson Labs. It's not meant as an all-inclusive, authoritative index by any means. It's meant more of a reference of what we have seen in the industry and what we would like to share with you regarding regulatory test procedures and other items to look for when performing particulate matter testing. The agenda for the presentation will be as follows. We'll start with a brief introduction for the concern regarding particulate matter on medical devices and in solutions, some basic definitions in the USP and other references, some sources of common sources of particulates on medical devices, a brief overview of establishing limits for your device or product, some available standards that currently exist, and then on to test methods and developing test methods, and also common issues that may come up during particulate testing or post-testing. First, the concern for particulates mostly has to do with the opportunity for particulates to create an immunological response in the human body. Health risk to patients is a very real possibility with particulate matter. It can come from, like I say, an immunological response, inflammation, or allergic responses. It can also occur from blockages, embolisms, strokes can also be a product of significant particulate matter, whether it's the particulate or the coagulation of red blood cells or other immunological responses. So particulates, depending on the nature of the particulate, can cause a great deal of problems. The size, shape, composition of particulates has a large factor in what type of response may occur, as well as the health of the patient currently. As I mentioned, particles can cause an immunological response, blood clots. There's a photo there of a cerebral artery causing a rupture from particulate matter or clotting, particle implantation. Ultimately, the body is not well equipped to deal with non-organic particles, and because of that, there can be a wide host of problems that can occur from particulate matter. Now, some basic definitions that we may use throughout the presentation that are commonly used in particulate matter testing. First, particulate matter, by definition, is regarded as mobile particles other than gas bubbles that are present on or arising from the use of a medical device or liquid. Particulate matter is matter in the vascular system which may cause an immunological response or intravascular coagulation or distal embolization. The body typically will target particulates for destruction like they would a bacterial cell or other foreign matter, but like I say, it's not well equipped to deal with it and to get rid of particulate matter, especially in large quantities or large particles. This is a micrograph. This isn't one we took, but of a particle looks like about 25 microns. Most particulate matter is similar to this, just chunks of nondescript matter, dust, just pieces of white or gray flakes, very common. These are a few that we've taken ourselves. Some are colorful in some ways. The one on the bottom left is red. Looks like there's a shade of orange or brown on that one at the top right, top left. But most are just, like I say, nondescript white or gray particulates. A fiber, fibers often come up as a concern to some regulatory as well as manufacturers. Because of the nature of fibers, they can be very long, but rather narrow. It's sometimes difficult to size fibers. It's not difficult to necessarily size them longitudinally, but sometimes a TAPI chart or other reference may be used when sizing fibers due to their odd shape. Also, the light obscuration counter sometimes has a difficult time sizing fibers, especially if it's trying to size it longitudinally and the maximum range of the machine may be too small to size a fiber accurately. The GMP's definition of a fiber is particulate matter with a length at least 3 times greater than its width. So there's an example of a fiber, red fiber. Sometimes it's helpful to try to characterize fibers and other particulates by color or size, or shape rather, in order to determine where in the process of either manufacturing, packaging, or handling a particulate may have been generated. The visible size range is typically regarded as about 100 microns. Some can see larger than that, smaller than that, and some can't see anything maybe smaller than 200 or 300 microns. But typically 100 microns is considered the visible range, and most of the time we're interested in particles below that range for subvisible particulate matter testing. The sources of particulates, common sources are often thought of as the manufacturing of devices. Obviously, there's grilling, there's molding, grinding, other manufacturing steps that take place during a device's production, and most particulates we find are generated during the manufacturing process during some of the investigations that we participated in. However, there's many other sources of particulate matter. Of these 9 that are listed here, some of the largest contributors we've seen is packaging, as well as shipping and distribution there on the right. Those 2 tend to be a large contributor. Shelf storage and sterilization can also contribute. Typically, it's good to include those in the final product before testing to see if maybe that's a common source of particulate matter. We've participated in quite a few investigations in which we've tried to trace down, working with our client, as to the source of particulate matter. And we've found that all of these can have an impact. So it's important to look at all aspects of the manufacturing process, from the raw materials all the way to the packaging and handling at the end, to determine which parts of that process may be contributing particulate matter to the device. Specific to the manufacturing process, we found that it's a good idea to try to control the manufacturing space, the environment, to look at the particulates generated during manufacturing, to possibly identify those particulates if possible, if they're significant, to see where continuous improvement might be appropriate. Also, as far as controls are regarded, to control the number of particles that are found on your final device, it's important to obviously minimize the amount of particulate matter that is available or generated during the manufacture of a device. Environment can contribute significantly also to particulate matter. We don't manufacture anything here at Nelson, but we do test our clients' samples, and so environment is a big deal to us. Airborne particles are very common, obviously dust, and which is probably the most common type of particle that we observe. Using HEPA filtration, as well as reducing the amount of high velocity airflow in the area, is a couple of good ways to minimize the amount of particulate matter that may be subject to the area. Also, reducing or controlling the amount of personnel in an area has also shown to significantly decrease particulate matter in most environments, as personnel tend to be the largest contributor of environmental particulate, clothing, skin, things of that nature. So here at Nelson, we say no to dust bunnies. We're very aggressive about keeping dust and other contaminants out of our laboratories. Specific to the particulate lab, we use a Class 7 HEPA filtration system to control our environment, and then of course a Class 5 for testing, which is required by the USP. Regarding personnel, as I briefly discussed, hair, skin, aerosols, all things of that nature, cosmetics can also contribute to particulate matter in the environment. Yep, as we talked about, skin cells, makeup, clothing. Clothing's a big one. Fibers from clothes, especially if clean room gowns aren't being used, which in many cases they may not be. Clothing, lab coats can contribute. Some controls that can be established, short of obviously, classifying an environment, is appropriate gowning, hand washing, considering restrictions on the amount of employees or personnel in an area, and those that may be ill, have coughs or colds, skin conditions, that type of problem. Particulate monitoring, we do particulate monitoring here at Nelson just in our own areas. As a manufacturer, particulate monitoring is also a good way to keep an eye on the environment in which your device is manufactured, and to also help track down during investigations where particulate matter may be coming from if you're seeing a significant amount on your device. Here at Nelson, we've initiated a monitoring program in our particulate lab and other parts of our laboratory. As a manufacturer, it's not a bad idea to also establish families of devices or raw materials and where those are located, establishing action plans and alert limits for investigations, and keeping a library of contaminants and lubricants, things of that nature regarding equipment. We've often found this to be significant. We've found oils and different manufacturing contaminants that are specific to the machinery used on some devices, and during investigations has come back to find that there may be some equipment that may be contributing particulate matter, whether it be in the form of actual solid particulate matter or in some cases oils or other lubricants that don't necessarily qualify as a particulate but can sometimes cause false positives with our particle counter. Now, establishing limits is a bit of a gray area currently in particulate testing because there are no set limits for medical devices, for most medical devices. Limits do exist for small groups of devices, as well as for liquid samples via the USP 788, which we'll go over briefly. But for most cardiovascular devices and implantables, limits do not currently exist, and so they must be established by manufacturers or other standards or specifications adopted to your device. So TIR 42, which we'll also go over briefly, gives some details regarding establishing limits, and we'll touch on that in a few minutes. There are some particulate matter standards that do exist. USP 788 is the most common for particulate matter in injections and parenteral infusions. Anything that's injected into the bloodstream or tissue usually has to pass 788. There are very specific limits for both small volume injectables and then large volume, which are things such as IV bags and larger volume fluids. 789 is specific to ophthalmic solutions. It's a little bit more liberal than 788 because it's going in your eyes rather than in your bloodstream. And while it doesn't sound like you'd want too many particulates in your eyes, it's, I suppose, not quite as bad to have them in your eyes as it is in your blood. So that's a little more liberal than 788. EN 45502 is a standard specific to devices, but it's specific only to active implantable medical devices. Most of these will include pacemakers and different accessories that may go with those. We test very frequently to that standard, but again, it's specific only to those types of devices and not to others. So while it sometimes has been adopted to other devices, it's not usually recommended unless your device fits into that category. There are a couple other obscure ones. ISO 8536-4, harmonized with 1135-4, specific to gravity-fed infusion devices. Again, a very small niche of device. But if your device does fit into one of those two, or there's a couple others that exist, there's an AT6 1996 that's for analog, or let's see, that's for I can't remember the exact device. That's for recollecting blood during internal bleeding, basically auto transfusion devices. That's what they're called. So this is why you should put everything on your PowerPoint. But most devices do not have a standard, as I discussed earlier, and so we'll get on later into how to set limits for those devices. So if your sample device falls under an established standard, then it's obvious that you should follow the standard. For these two types of devices, the EN and the ISO are good references. But when your sample or device does not fall under a standard, which is most cases, then a test method needs to be developed, and we'll go into more detail. But ultimately, testing of any device, when testing for particulate matter, should closely mimic the clinical use of the product. This has been recommended by most regulatory agencies and is also recommended by the TIR document. Testing should also include accessories and supplies used clinically. The purpose of this is to see if there's any sloughing or interaction between those accessory devices and the test device. The test method should also be validated in most cases to determine if the particulate recovery from the sample and any model or supplies that are used is adequate. In some cases, the particulate matter that's generated may be retained within the model if a model is used during testing, or other accessory devices. Most regulatory likes to see a significant recovery of particulates, usually over 90% of what's subjected into a model or accessories during testing. Some typical methods for determining particulate matter on devices. Historically, the small volume USP 788 specifications have been used, and the method in that standard referenced for testing of devices. However, in the last couple of years, this method of adopting the 788 to devices has been less accepted by most regulatory agencies, including the FDA. It does depend on the nature of the device. In some cases, implantables or smaller, less invasive devices may still be adequate to be tested to the 788 specification, but in most cases, it's recommended to come up with a custom protocol and not to rely on 788 for testing of a device. Using historical data of the device along with comparative data, which we'll go into more detail on both of those, of similar devices is also an excellent way to determine a method and particulate limits for your device. Often this is comprised of taking a competitor device that may already be on the market, performing testing on that device similar to the one you're testing, and then comparing the particulate matter data to both samples, the one that's currently on the market and the one that's in development, to see where you're currently standing. And the justification for this is probably fairly straightforward. That is that if a device or product is on the market and is shown to be relatively safe clinically and contains the same or more, or at least similar amounts of particulate matter to the device that's being tested, then it can be justified that it should be relatively safe. It is helpful to have other tests combined with these when using these methods of comparison. Endotoxin testing, TOC, total organic carbon, tests like that can show biocompatibility are also good when using a comparative method, as particulate matter is just one of several tests, as you know, that have to be conducted on a medical device. So discussing the TIR document briefly, it's TIR 42. It's an AAMI standard, or an AAMI reference. It was released December 13th, 2010. As Mike shared, Tanya Morris was part of the committee that helped develop and write the drafts and ultimately the final for this document. It's an excellent document. It's a must-read for anyone that's involved in particulate matter testing of a medical device. It provides a great amount of guidance regarding particulate sizes and shapes, more on the test method setup and validation that we briefly discussed previously. A lot of what was in this presentation so far, such as common sources of particulates, came straight from that document. Recommended sample handling, recommendations for setting limits, action and alert limits, as well as some particulate identification techniques in the case of investigations is also discussed in this document. So definitely an excellent document to read if you are involved at all in particulate matter testing for a device or even a liquid. It contains a lot of good tips and technical guidance regarding particulate testing. The FDA has also issued some guidance specific to PTCA catheters. This was issued last year. Although this is just for these types of catheters, this document also contains a great deal of valuable information, especially since it did come directly from the FDA. While the FDA was participated on the TIR document, this document is straight from the FDA. So it contains some redundancies with the TIR document, but is specific more on exactly what should be done in testing of some devices, including the PTCA catheters, of course. Includes measuring the quantity and size of particulates using simulated use, conducting testing on the finished product. This is often a big hang-up with some clients where they may not test the device post-sterilization or aging. It's definitely recommended to test post-sterilization. We haven't found sterilization to typically increase particulate matter very much, but it's one thing that can be done to decrease arguments. So as any of you who have been involved with submissions to the FDA, it's always good to get rid of as many of those arguments as possible. Sampling plan information. This is important concerning lot variability, different sizes. We've seen some hang-ups considerably with stent systems where there may be several different sizes, even material composition of the stent itself. It's always good to have several different sizes and types of your device if there are any type of variability in the size or composition of the device or product. Inclusion of accessories and model or a tortuous path. We'll talk a little bit more about the tortuous path, but basically inclusion of devices or models that help model the clinical use of the product. Also worst case scenarios and testing to a worst case scenario. And then information regarding the test method validation, some specific numbers that are given, which I have later in this presentation. But like I say, they like to see about 90% recovery on those particulate sizes. Now, speaking a little bit more of comparative and historical data, obtaining data from multiple lot sizes and types of a product, as well as competitor designs already on the market, have a great deal of value in doing particulate matter testing. Mostly because you're getting data, a significant amount of data from your device as well as competitor devices, which can show justification very well to the safety of your product, which is ultimately what the point of the submission to regulatory is. Some of the benefits of comparative and historical studies is that they can provide a measurement of how clean your device is compared to others. This isn't just for purposes of regulatory, but also to give you an idea of the nature of the sample, product design, and kind of gives you a warm fuzzy. It's nice to see that bright look in the engineer's eyes when they see that their device is significantly lower than those that are on the market, so it's a good feeling. Also, it can provide original data that can be leveraged for regulatory approval. Historical data can allow for trending of manufacturing, handling, and packaging cleanliness. So even beyond the regulatory process, we have many clients that test routinely their products just to be able to show trends, see if there's any out of spec or out of trends going on with their current manufacturing process and be able to monitor that process more closely. In the case of stability testing, it allows you to observe particulate level changes associated with aging as well, which can be significant. Oh, let's see. I pushed the right mouse button instead of the left. There we go. Okay, test development is typically required when testing devices, not so much with liquids, but specific to devices. Again, mimicking clinical use is of utmost importance. The FDA and most other regulatory agencies will really scrutinize the test procedure at hand. They'll want to know that everything in the test procedure follows very closely with the IFU of the product. Use of a model that mimics the vascular structures that the product will be subjected to is very common and usually recommended. Submitting samples to physiological or clinical conditions as far as temperature, saline, and also sufficient manipulation or movement that may take place clinically by the physician during use of the product. Worst case scenarios are also a good idea. Speaking of sufficient manipulation and movement, in many cases we will reuse essentially the device or not so much reuse, but retract in advance, say if it's an introducer, a catheter, a guide wire, something of that nature, several times to mimic if the physician were to be struggling to get that product in the right position in the vasculature. But also sonication is another addition to worst case scenarios. That's above and beyond what we normally will do for extraction, but that will definitely remove pretty much all particulate matter off of a device. So that's another option for a worst case scenario. Now the test method validation is an interesting piece of procedure that's been introduced in the last several years. Here at Nelson we've performed a great deal of these validations. Validation might be a fancy word for it. I typically call it a particle recovery test or a particle recovery validation. This is typically only required when a model or significant amount of supplies or accessories are used in the test. And the purpose of this, as described earlier, is to make sure that the particulate matter that's generated during the test is sufficiently removed and extracted from any accessories and models that are used so that they may be analyzed on the particle counter or microscopically. In any cases where particulate matter is harbored in these, it's essentially the same as the particulate matter being harbored in the bloodstream, which isn't really accurate if you're only getting, say, a 50% recovery in your test and showing rather low numbers, but in truth, twice that amount of particulate is going to be subjected to the patient. We normally use what's called the spike and recovery method, where we'll inject the model or accessories, essentially the test system, with a known quantity of particulates. We'll normally use Thermo Fisher's NIST traceable particle standards, previously Duke Scientific. Many of our sponsors have used those before for calibration purposes or running validations of their own. They are specific, they're at specific concentrations, usually around 3,000 counts per milliliter. We will perform the test protocol as written, minus the sample, of course, and then collect and analyze the sample that's retrieved, sample referring to the fluid sample coming from the model and accessories used in the test. We can then compare the observed counts versus the known counts and come up with a comparison between what was spiked on the sample compared to what we started with. As far as numbers are concerned, the FDA in their guidance document has historically preferred to see greater than 90% recovery for the 10 and 25 micron sizes, the USB sizes, and at least 75% for larger particle sizes. Those have been defined as larger than, equal to or greater than about 50 microns. We usually use anywhere from 50 to 70 microns for that larger particle size. And the larger particle sizes are interesting. There isn't typically a specification for larger particle sizes, nor are you typically required to come up with one. But it is a bit of a red flag when there's a significant amount of these larger particles, as a single large particle can cause more trouble than multiple small particles. Now, addressing the USB 788, we frequently get questions from many of our clients as to whether or not they can adopt the 788 to their device. Much of this has come from historical use of the 788, and some of it has come from just the fact that the 788 is the most common standard used in particulate matter testing. But some things to keep in mind is that the 788 was written specific to injectables and parenteral infusions, just liquids. Sometimes it is commonly used as a green zone. Even here in the lab at Nelson, we typically will regard a device to be rather clean if it's below the 788 specifications, but it's not really designed for medical devices. The standard is designed only for liquid samples, and therefore it doesn't contain any instructions, no procedural details as to how to test the device. In addition, the FDA has often mandated that meeting this standard, even if your counts are below the 788, does not necessarily mean that a device is acceptable, that a device has acceptable particulate levels, though it usually can be leveraged as justification. It's not difficult to use the 788 as some sort of justification that if a solution has X amount of particles and the device has less than that, it should be safe. But it's still not a good idea to depend on that justification on its own for approval from any regulatory agency, maybe as some type of backup or a scientific rationale, but not on its own. Now, quickly, some of the specifications for those who may not be familiar with this USB 788, there are about thousands of USB, well, I don't know about thousands, but many USB references. So we don't know all of them, but in particulate matter, these are the specifications that are outlined for USB testing. There's the light obscuration method and then the microscopic method. The light obscuration, as you can see, is significantly higher. It's twice that of the microscopic. The reason for that is I don't know entirely, but after several years of particulate matter testing, it is typical to see higher counts with the light obscuration method. False positives can sometimes creep up with light obscuration, and it is more sensitive. So the counts are higher for light obscuration than microscopic, and so thus the higher counts on the standards. And keep in mind that the light obscuration and microscopic only request the 10 and 25 sizes. It is typically recommended to test a larger particle size with devices, like the FDA recommended greater than or equal to 50 microns. These are the specifications for large volume. Those previous specifications were for small volume. Small volume are more frequently adopted to devices because it gives it as counts per container. Large volume gives it as counts per mil. Counts per container can easily be adopted to counts per device, but counts per mil is a little bit more sticky. It's in some cases where the device is used for preparation of a large volume injectable, such as IV bags or drug mixing bags, the large volume specification might be appropriate. But in most cases, the large volume spec is not used at all or even attempted to be adopted to any type of device. Some other comments about the 788. Like I say, adopting the USB 788 to a device has historically been acceptable, but we have noticed this shift with the FDA, where they may accept data within the USB specification depending on your reviewer, but they still want to see historical data, comparative studies, and method validations. If you just simply present them with data that's below the small volume specification for the 788, I wouldn't expect to be able to just receive approval based on that alone. Here at Nelson, we definitely do not recommend adopting the 788 in its entirety to a medical device. Apart from the fact that you pretty much can't do it because there's no procedure, there's a very good chance, almost certainly, that it won't bode real well with most regulatory agencies. Now some information on some method advantages, kind of switching gears a little bit. The light obscuration method is the preferred method by the USP, and most regulatory also likes to see the light obscuration method used first with a device, or with any testing for that matter, in particulate testing. Probably the most important reason for that is the repeatability and objectiveness of the light obscuration method. The light obscuration method uses a liquid particle counter, which is a calibrated piece of equipment. It's non-biased, it's repeatable, and so regardless of who is testing, regardless of the technician, you're going to get pretty consistent results from the light obscuration method. Also, it's able to quickly analyze large quantities of samples. So regarding the convenience of it, it's also a little more convenient. It's less expensive. We definitely charge less for the light obscuration method as it's a lot quicker than the microscopic. Fluid can be further analyzed as well. If you have a sample of fluid, say 100 mils, the light obscuration method only requires 25 if testing to the USB 788. So actually only 20, I should say. We typically ask for 25. It draws 20, but we usually want a little bit more than that so we're not sucking up air at the end there. But yeah, only requires a small amount of fluid for analysis relative to an entire sample. Also, it can count very small particles down to around 2 microns or even smaller. Typically on a light microscope, you can only see down to around 10. Some can see 5, but accurately counting particles below 10 is pretty much out of the question with the light microscope. The microscopic does have its advantages, though. It has some ability to identify particulates, especially if you're looking for a certain color. If, in many cases we may be looking for green or blue particles. If we're looking for coatings from catheters or guide wires, it's handy to be able to see the colors and the shapes of the particles. Light obscuration only gives a count and a size. It doesn't give any kind of description of the particles. The microscopic also is not impacted by any oils or surfactants, bubbles, any sort of biological particles, proteins, or anything of that nature. We test quite a few biological products here, plasmids and different vectors and things that may cause problems with the light obscuration counter, or the liquid particle counter rather. And the reason that those compounds cause problems is because the liquid particle counter essentially is kind of like a culture counter in a way. It has a beam, a laser beam. It sounds a little futuristic, but for lack of better words, a laser beam that is projected, and a sensor on the opposite side of the apparatus that then detects blockages within that from that laser. The beam that's shined through the sample. And in cases where a particle comes in contact with that laser, it diffracts and is accurately sized and counted. Unfortunately, oils and bubbles and these other compounds may also diffract the laser even though they're not true particles and be detected as particles. That's really the main purpose of the microscopic method nowadays, is to confirm counts from the light obscuration method and to use in the case where your sample may not be compatible with the light obscuration method. But even in cases where you may know that these compounds exist on your device or your sample, it's still a good idea to have that sample run using the light obscuration method to see if those compounds will cause a significant interference. And if they do cause interference, to have justification for going to the microscopic method. The FDA specifically will still like to see data with the light obscuration method, and then that same sample compared to the microscopic to show that, here's very, very high counts with the light obscuration, here's very low counts with the microscopic. We know our compound has X compound, we know that our sample has, a great deal of silicone lubricant on it, for example, and that's probably what's causing the interference. So they like to see justification like that. It's recommended to do the light obscuration first and then go to the microscopic if you have problems. The light obscuration, well, we kind of went over the disadvantages briefly, but yeah, colloids, surfactants, gas bubbles, all that can cause a problem with light obscuration. There's no options for identification. It is rather expensive equipment. If you'll be doing a great deal of particulate testing in-house, it may be worth the investment, but it's also a maintenance investment. Typically, it's a good idea to have it calibrated per manufacturer's instructions, which is typically semi-annually. That's rather expensive also. It does require a minimum volume also, whereas the microscopic doesn't. You can test a very, very small volume with the microscopic. The microscopic is somewhat subjective. Counts can differ from one technician to another. The labor expense is high with microscopic. It takes quite a good deal of time. We've spent as much as 30 minutes on a single slide. And so, if you've got multiple samples, that can get expensive from a personnel standpoint. All the extract is typically consumed in a microscopic test. You can test just a portion of the sample, but normally, per the USP, the entire sample is typically consumed in the test, so you wouldn't be able to do a further analysis except for the particles that were present on the filter. It does require a greater amount of time, and it's difficult to examine small particles with the microscopic method. Ultimately, here at Nelson, we recommend consulting. Consulting with us is good. Also consulting with the FDA, getting them involved early. Anytime during the formulation of your procedure, your test procedure and protocols is a good idea. In many cases, this can save you a great deal of time and money. We've had multiple times where we've had our clients come back to us and say, well, we have this additional size we didn't include, and now they want, the FDA wants this size tested. Now we've got to spend all this extra money and time and delay things. That's just one, scenario of hundreds, literally, that we've come across. We do our best to try to help you cross all the T's and dot all the I's, but it's a good idea to consult with multiple agencies and institutions to try to make sure that we have everything taken care of to start with before we have all our results and then find out something was missed. And some common issues, these are probably, these 5 right here probably encompass 90% of the problems that come back to us post-testing when submitting a 510K approval to the FDA. The first is typically not including critical steps or equipments or variables within the test protocol. This is like I discussed in that scenario just before, say there's a certain size or maybe a certain time point that wasn't included in the test or that wasn't tested. That can be significant depending on the situation and the number. Also not testing all the sufficient varieties of the sample if there's different sizes or types. Now it's not necessary to test a great number necessarily. Particulate testing, if done by a contract laboratory, can sometimes be expensive if there's multiple samples. If we're talking 50 or 100 samples, it's not typically necessary to test so many samples, but say having 3 or 5, say maybe 10 if you want to get fancy. But testing a good amount, but not necessarily many samples. But just having, here's 5 samples of this size, here's 5 samples of that size, here's 3 of this particular, type of stent that is a copper base or a chromium base or whatever. I'm not familiar with all of the materials used. Your engineers are probably laughing at me, but yeah, basically, testing all the different varieties, not necessarily testing a lot of them, but just having a few of them in there. So you can take that data and say, oh, here's this variety, here's that variety. Definitely a good idea. Not conditioning the samples prior to testing. This includes packaging and stability or aging. In many cases, we've seen where our clients have not done sterility. I put stability. That's also true, but sterility testing prior to sterility prior to testing. And that's had a hang up with the FDA. We found that sterility rarely impacts particulate matter, but that the FDA just likes to see it. It can have an impact, depending on what sterility method is used. Heat can sometimes have a higher impact than, say, EO, but they like to see, a final product because they want to see what's going to be subjected to the patient, and that's understandable. Also, not enough data points, insufficient units tested, or an insufficient sampling plan. I said earlier you don't necessarily have to have a ton of units, but you should have a good amount. I wouldn't submit 3 units to regulatory, 3 points of data and expect it to fly. You're probably going to want 5 or 10 or 20. It's a sufficient amount, a good statistical number. And most sampling plans in most major manufacturers are pretty good, so that hasn't typically been a problem, but that's why it's number 4. And then number 5, the last, really not common, but every once in a while, if we get low particulate recovery results from our validation, that can be a hang-up. If it's much below, 85, 80%, I've seen regulatory agencies take less than 90, but if you're down around 80 or below, you're probably going to have some hang-ups. So if we're approaching a little too low on those recovery results, if a validation is necessary, then we may want to go back to the drawing board and see why we're getting such a low recovery. As far as contact information, both myself and Tanya Morris are always available. Those are our contact numbers there. You can always get a hold of us here at Nelson Labs at the sales department. They can forward any information or questions to us that you may have. Tanya is very involved in particulate testing. And then we also have another study director just recently. His name's Steve Lamb, and he's in charge of the microscopic testing. And so we have two full-time study directors working in particulate testing, and then Tanya Morris also. So between the three of us, we'd be happy to help with any questions you might have. We're always happy to answer questions, even if you're performing testing in-house and maybe just want us to analyze your fluid. We do that very frequently also. So that's certainly acceptable by us. But yeah, please let us know if you have any questions, and we'll be happy to help. And I'll go ahead and hand it back over to Mike. For anyone that has questions, go ahead and submit those, and we'll do our best to answer those in the time we have remaining.
Thanks, Ryan. We appreciate your insights, and we've all gained some great information and knowledge today. We've given us a lot to think about. We have had lots of questions come in, so we're going to try to get to as many of those as we possibly can during this remaining time that we have. If we don't get to your specific question, you can always, like Ryan said, you can always contact him or Tanya or a member of our sales department, and they would be happy to assist you with this. But I will turn the time back over to them, and they'll be able to start getting through some of these great questions that we've had come through.